Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate datasets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to this very special episode of the Genetics Podcast. This is our 200th episode, and for this very special occasion, we wanted to do something fun and different this time. So we're going back in time, and we're pulling out some of our favorite clips from some of the early episodes. All of these will be in the first 100 episodes, so for many of you that may have found us in the last couple of years, this is probably something you haven't heard yet unless you've had time to go back in time. And even if you have, we picked these episodes and these clips from the episodes as things that were either amazing predictions about what might come years in the future that ended up being true, or timeless, really great conversations about some enduring and important themes or topics in our industry. So we hope you enjoy, and thanks for joining this very special episode. All right, we have some very great clips today from 4 different past guests. The first one is Eric Topol. The second one is Laurence Reid. The third one is Vineeta Agarwala. And the fourth one is Daphne Koller. So jumping into the first one, Eric Topol is a household name for many of you all. He's got a very popular Substack, and he's been an incredibly prolific researcher in genetics and genomics more broadly. When I think about guests that made predictions that were ahead of their time, he was one of the first ones that I thought of, because way back in episode 10, July 2019, I asked him what he thought healthcare might look like a decade into the future. And there were some really incredible predictions that I think you'll be hearing today, in particular around AI. So listen closely through the lens of everything we've experienced now. Try to take yourself back more than 6 years ago now to July 2019, and I'll be back with you in a minute. You have I think, a pretty incredible vision for the future of healthcare and medicine, and it involves artificial intelligence and machine learning. But the way you see it is, I think, actually very different from what many people think. So I was wondering if you could maybe just paint a picture for us now of how you see the world of medicine and healthcare being different a decade from now. What will life be like for the typical patient?
Well, it comes down to that there's immense data for each of us. And it spans not just the traditional medical data that might be in a record or records that are dispersed in many different places, but it has sensor data, our genome, our gut microbiome, our environment, and all these different layers of data. I mean, I haven't even mentioned some of the biology layers like the immunome, whatnot. So basically, that is transcending human capability of ingesting and dealing and analyzing such data. And that is going to be the future of medicine, where AI we rely more on machines to do that, to help us, to gather it all and crystallize it, distill it. And that will make for individualized medicine, which is very different than the way medicine is practiced today, which is essentially dumbed down and very shallow.
Right. What do you see as the kind of low-hanging fruits, or what are the first things that'll happen that will mark this transition? Or maybe some of them are already happening today.
Well, some are happening. Today, the first things that we're seeing are things with patterns like medical scans that are now getting pre-read by a machine before the radiologist sees them. And that is great, because over 30% of scans have a missed finding, a false negative. So that rate can be dropped much lower. And that's getting a lot of uptake. It's a positive thing. But we're also going to see that same sort of accuracy and speed applied to many other specialties. Pathology, dermatology, ophthalmology, and then most recently the randomized trial in gastroenterology, that is colonoscopy to pick up these small polyps that could be precancerous at a rate that is extraordinary, that are missed by the doctors. So, this big improvement of accuracy and efficiency is welcome. We need that. The UK is leading the charge in certain areas. They're the first to make the voice recognition a reality instead of typing on a keyboard in emergency.
Really?
Yeah. So that's exciting because we're going to eliminate keyboards someday. And everyone talks about going paperless, and we've never really been paperless. We got to go keyboardless because it's the keyboards that are the common enemy of the people.
Right.
And patients and doctors. And that is the first step of getting eye-to-eye contact and presence and, real human interaction.
I thought it was pretty interesting to hear how accurate some of Eric's predictions were. In particular, for me, actually, the discussion about digital scribes and how these can give doctors more time and more human connection with their patients, I think is a really important perspective in the AI boom heyday that we're in today. For me, more broadly, I can see AI as being embedded across nearly every layer of healthcare and precision medicine. But I also think the next 6 years is going to bring way more change even than the last 6 years has. For me personally, I'm most excited about the way AI can be used to combine data sets together to create a clearer picture of us, of our risk, of our health, and can drive a new wave, hopefully, of AI diagnostics and risk prediction. We have a lot of work to do. I think AI is only as good as the data sets that it has access to, and we don't yet have a clear patient-centered picture of all of us. And we know that the data sets that we have today are biased, and we have a lot of work to do. But nonetheless, I'm pretty optimistic about our future here. This next clip is from May 2022, and it's about a trend that, as you all know, if you've listened to this podcast, I'm really interested in, which is gene therapy. And back in 2022, I spoke with Laurence Reid, who at the time was the CEO of Decibel Therapeutics. At the time, Decibel was just preparing to file their IND. So they hadn't even launched their clinical trial yet, but they were planning and getting ready to kick off a gene therapy targeted at a particular genetic form of hearing loss called otoferlin-related hearing loss. In this episode, we went deep into inner ear biology, how to build a gene therapy company. And for me, this was pretty amazing because years later, Decibel went on to be acquired by Regeneron Therapeutics, and they hit front page news, in major publications when their clinical trial was found to have restored hearing to a number of children with rare and previously very difficult to treat forms of hearing loss. This study has continued, and they've continued to build momentum. Now for older and older children moving into adults that are getting this gene therapy, it's been found to be safe, and it's been found to be effective. So hearing Laurence talk about this back in 2022, and really just 4 years later, seeing this have a major impact on patients was a pretty cool moment for me. So I hope you enjoy this, and I hope this takes you back in time and gives you a little bit of perspective on, yes, our industry moves slow sometimes, but with a lot of focus, with the right people and technology, we can make a pretty big impact.
There are many genetic forms of hearing loss. I think gene therapy is going to be such a powerful modality, has the potential to be such a powerful modality for the inner ear, but you need to access it. So it's a complex set of drivers and variables that are going into how do we get this exquisite technology from a molecular biology perspective into the right place. And, we think the ear is a very exciting opportunity for gene therapy. And we really see the ear now as one of the vanguards of the gene therapy field. So, and of course, now we've still got a way to go in terms of proving that the various pieces that I just articulated, that we can join them up in clinical studies, which we'll get to in the next few years, and incredibly excited to do that. So there's a landscape there of different areas where we think the first product candidates are going to get developed. And, in terms of picking amongst them, the way we think about it, obviously one is trying to, influence and try and change the lives for as many people as possible. But also there are, of that 90 to 100, there are some where children who are born with a profound lack of hearing, the impact is probably going on, in utero, and their ear hasn't developed properly. And in terms of postnatal gene therapy, it may be that those are not accessible to us, in the immediate future. So we try and focus on ones where there's a decent sized patient population that we can imagine, running a trial and having ultimately a viable product with a significant number of lives to be influenced, but also that our understanding of the biology of the ear, despite the genetic, appears to be functional such that if one then returns the gene that is mutant from parents, that you actually think you've got a reasonable probability of success that the child is then going to emerge with a more normal form of hearing. And so you have to think very carefully about targets from that perspective. Otoferlin, where we and others have chosen to start, seems to really tick that criteria. And it would appear from both human studies and animal studies of genetic models that the ear is functionally intact, except it lacks this one specific gene that functions in the hair cells of the inner ear. And we and others have done extensive rodent model studies where we can rescue the phenotype in a pretty convincing way, in an animal model, and actually achieve that quite a long time after the birth of the animal, which gives us hope that therefore we're going to be able to do that perhaps over a range of ages of children.
Great. I hope you enjoyed that one. And as I mentioned before, Decibel went on to be acquired by Regeneron after we recorded this just about a year later. Earlier this year, in 2025, Regeneron presented some of their results from this trial. Out of the 11 children who received the treatment and had at least one follow-up, 10 showed measurable improvement in hearing loss. And there's been recent publications as well about how children's hearing compares to those with the same condition that had received a cochlear implant, for example. And I think this shows the potential of gene therapy. We still have a ton of work to do, and it's by no means a finished technology, but this gives me a lot of hope for patients who didn't have an option except for cochlear implants prior to this gene therapy success and now have many more options on the table for them. This next clip is one of my all-time favorites. It's episode 40 from July 2020 in the heat of the pandemic with Professor Sir Rory Collins. And looking back on it, at the time, it was obviously clear that the UK Biobank was just an incredible force in driving innovation in our field. That hasn't changed. If anything, it's just continued to compound with all the new multi-omic data sets that they've been bringing in. And at the time, what I really loved about this was just hearing from Rory about the vision and the perspective that it took to get the UK Biobank started. They really didn't have a hypothesis. They knew that if they built it, people would come and would generate all these amazing things. And I'd like to think that it's surpassed even Rory's expectations at this time. But if you work in this field, you probably spend a lot of time thinking about UK Biobank data sets or working in ways that are influenced by the work that's been done there. So I hope you enjoy this episode, which is one that I think is just timeless.
I remember we went to the American Society for Human Genetics and said, look, we've got genotyping on 500,000 people. And people were going, you have what?
Wait a second. Yeah.
We suddenly saw this big increase in research use from the US and other countries. And I then got a call from somebody from Regeneron, Arisparis, who said, you've got this resource. I mean, can we get access to it? And I said, yeah, it's available to academic and commercial researchers on the same basis. And he said, yes, but can I really get access to it? And so one of the big obstacles has actually been persuading people that it really is available.
Right. There's no tricks. Yeah. What are the things that have either surprised or excited or interested you the most that have come out of the project?
When we had to go back for our renewal, UK Biobank 3 years ago or so, I was asked, what had UK Biobank produced that it had been set up to produce? And my answer was nothing, because it's a prospective cohort. And the last 10 years or so have been about building it. So I really feel that it will be the next 5 to 10 years when it starts to deliver the really major findings for which it was set up. Having said that, I think the things I've found really interesting have been how you do things at scale, has been from a kind of practical perspective, has been really interesting. I think the value of creating a lot of data and then putting it out there and letting people's different imaginations work on it. And again, the one thing I would really like to do over the next few years is to increase the numbers of imaginations that are applying themselves to the data. We know it's being used a lot in the UK, in parts of Europe, in North America, but there are a lot of other places in the world with equally imaginative scientists that maybe don't have the resources, the facilities we luxuriate in. And so one of the really nice parts of the whole genome project is that we've been funded as part of that to solve the problem of the scale of the whole genome sequence data. we'd been funded to get a data analysis platform set up that would allow researchers to work on the sequence data, to go to the data, because it's now too big, or it will be too big, for the approach we've taken up to now, downloading the data to researchers. And I think that that's going to massively democratize access to the resource, because researchers who don't have a big computer all over the world will be able to go to the data. And in fact, we announced the outcome of that tender. So DNA Nexus with Amazon are going to support this platform. And as part of it, Amazon are going to provide $500,000 worth of free compute every year for researchers in low and middle income countries and for students. So that, I think, will be a way in which we can get very many more people using it. But the thing I've learned is just how much can be produced by having lots of different minds with lots of different, you could say, points of view, prejudices, approaches applied to the same data. You get quite different things coming out of it.
Yeah, and it sets up a system where the best idea wins, right? It's no longer that the data is a scarce resource, somebody's sitting on it and just continuing to publish once every few years. You really have you reverse the situation where you have as many smart brains pointed at this vast and multifaceted data set as possible. And then the level of creativity is just through the roof, right? I mean, I think the explosion in understanding of polygenic risk scores, for example, that in particular, I can't imagine what that particular part of the field would be like if the UK Biobank didn't exist, because we'd be probably still stuck in case control studies that are in large part, there's a degree of power that's held by the group of people that created that cohort in the first place. But something like this has the potential to, well, it really already has, but as you say, going forward, even more potential to just level the playing field in terms of data access.
Yeah, I mean, the polygenic risk scores is an interesting one, and one where you can imagine it's going to have quite a rapid practical value. So the idea that you can find, 3, 4, 5% of the population that have a risk equivalent to a single gene disorder, be it familial hypercholesterolemia or BRCA1, BRCA2. And I think Peter Donnelly has said if you take 16 diseases, then you can find a quarter of the population that is in the top 3 to 5% for one or more of those diseases. Well, that means that a genotype is going to find a lot of people where you might well be able to do something useful for them. So you're a 30-year-old, but you're equivalent to FH on a polygenic risk score. You think about taking lipid-lowering therapy. If you have equivalent of BRCA1, BRCA2, again, you might be thinking earlier age of screening. And of course, the other extreme, the people who are at the very lowest risk, you might have women who are screened either much later or never for breast cancer. So I can see that being implemented into healthcare quite quickly.
All right. I hope you enjoyed that. One of the things that stood out to me as well, that just listening to it again has got me thinking about it, is Rory's commitment to open science and how much he has been committed to that since day one of the program. So I can't stress this enough, how impactful the UK Biobank has been to science globally. And I think it's been the precursor to many other impactful projects that are coming down the line, like Our Future Health in the UK, who we've interviewed previously on the podcast, the All of Us program in the US. And I think many, many more are going to see the value of not just this open science approach, but also public-private partnerships. They've done an incredible job at attracting investment, not just from the government, but also from the private sector. And I think we all win together when we bring these powerful forces. So thank you again, Rory, for your visionary leadership on the UK Biobank. This next clip is another AI pioneer that I think was years before her time. So Daphne Koller was also one of my favorite episodes, episode 83, back in August 2022. Daphne had a vision that from the beginning of the company, they wanted to build something that built AI and machine learning into the drug development process from day one. And we talked a lot in this episode about why she chose to do that rather than, for example, going to join a large pharma that has huge amount of resources, and she may be able to build that kind of system internally. So obviously there are so many ways to do this, and there have been many large pharma that have been successful at taking this kind of approach as well. But I just thought the way that she broke down so long ago, the vision here to build basically a feedback loop between the lab and the machine learning models that they're building was just really exciting. And this goes back to 2016, the vision for this company. So clearly a decade ahead of her time in terms of the vision for an AI and machine learning driven drug discovery platform.
What I saw around me, and this was in 2016, 4 years after the machine learning revolution, and seeing that machine learning had been transforming the world around us, but not really having that much of an impact in the life sciences. And when I asked myself why that is, it's because there's not a lot of people who speak both languages. By and large, machine learning people don't know very much about biology at all, and most biologists are certainly not trained in machine learning. And so, and that's a chasm that makes it very hard to have a meaningful, deep application of machine learning methods to life science data with a handful of people who can really make that connection. And so I felt that the impact that I personally could have would be much larger if I went and built something that probably would not get built if I or someone like me didn't do that, versus just continue on the trajectory that we were on with Coursera, which would likely continue even if I left. And so that was the point when I didn't quite know how that impact would translate. I had been out of both machine learning and science for about 5 years, so I didn't even have an idea of what I wanted to do. And I was fortunate enough to connect with Art Levinson, who was the CEO of Calico, previously the CEO of Genentech. And so that was an opportunity to work with a truly great leader, and I was really inspired by that. And so I went and spent 18 months at Calico and realized that ultimately I was not a biologist. I was still a computer scientist, a machine learning scientist. And so what I wanted to do was to build a platform that would help solve biological problems in a very different way, and specifically transform the way, hopefully, in which we design and develop medicines. And it didn't make sense to build that within a company that focused on a specific biology, the biology of aging. And so that led me to depart Calico after about 18 months and form insitro, which is 4 years old this week. So we learned that causality is critical. We learned about the incredible amount of information that exists in high content data in subtle patterns that people just can't see. That has turned out to be something that we've repeatedly shown across multiple data modalities, both cellular and human, that there is way more information in histopathology than even an experienced pathologist, for example, can discern. Same is true for cellular images and so on. So that's one thing that we've learned. We've learned about, as one considers the tools and protocols that we use in the lab, that if you want to apply machine learning to high content data, the reproducibility and error parameters of the tools that you're applying are absolutely critical, because the more advanced the machine learning that you apply, the easier it is for the machine learning to get confused at things that are sort of artifacts and batch effects and latch onto them and make incredibly accurate predictions that have absolutely nothing to do with the underlying biology. And so that has actually driven the choice in many cases in our lab of methods that are really where the selection is very much driven by how reproducible they are, how robust they are, how much can we protect against artifacts and batch effects. That also has driven a very big focus on automation, because one of the biggest sources of batch effects is when you have different humans doing the same task, or even the same human doing the task on 2 different days, when they woke up on the wrong side of the bed or whatever. And then similarly on the human cohort data, which is the other big data source that we use in our discovery work, really focusing on data sets that are large and well curated and well collected. And in that respect, the work that we did with the UK Biobank and many others, I mean, to me, the UK Biobank is an incredible resource that has driven so much insight across so many different aspects of human biology, human disease, and even just epidemiology, because it is so well collected and well curated. So the availability of such resources, I think, is absolutely critical to scientific progress.
This episode has made me think about a little bit differently again, listening back to it, is I think the changes in large language models appearing on the scene and what they can do and what they can't do actually shines an interesting light into what the future might look like for us in biology. The large language models have the advantage of having an incredible amount of text data from all over the internet. What we really don't have is something similar in biology. We have all kinds of disparate data sources, but we have nothing in terms of the depth and scale that we're talking about there. So one thought to leave you with is, how do we get to the scale of data that might look like something a large language model takes in today in biology to help us fundamentally change the way we understand how our bodies and other organisms' bodies work. The other way to think about it, though, is, can we create these small feedback loops? They use cellular models and tie those very closely with their machine learning models. In that kind of smaller loop, that you may actually be able to generate this level of scale of data in a lab or a series of labs that might look something like the data centers that we're building today. So it's got me thinking about how could we take this mindset of building a feedback loop between what we observe in the real world in biology and what we can get a model to teach and predict, and how can we do that in a way that gets to the level of scale and impact that we're seeing in some of the text-based models, and what impact might that have on biology. All right, closing out here, we have another great episode that I think has some lessons that are still incredibly important years later. This is episode 63 with Vineeta Agarwala from Andreessen Horowitz back in 2021. So Andreessen Horowitz is a venture capital fund. This is one of the few investors that we've had on the show, but Vineeta is also a physician and has a background in genetics and life sciences more broadly. So we had a really interesting conversation about where the future is going to be from her perspective. And this is what she does for a living. She bets on early stage companies that have a vision of what the future might look like. And her success failure is literally measured by how those companies do in the next 5, 10, 15 years. So I thought it was a really interesting conversation. I'm going to be back with you in a few minutes to close out once you've heard from Vineeta. One of your investing focus points, I believe, is on unique data sets. I'm curious if you could talk a little bit more about that, whether it's genomic or other kinds of unique data sets that you're interested in.
Yeah. So we look at data sets, I would say, in largely two categories: data sets that drive discovery and data sets that drive development, clinical development of novel therapeutics. And in both cases, I think we're just seeing the tip of the iceberg in terms of how companies are going to creatively use data to accelerate and make more efficient both sides, discovery and development. On the discovery side, that could totally mean what we just talked about. It could be a company that's set up extraordinary access and know-how in mining large global population biobanks. It could be a company that's kind of crawling the UK Biobank, all day, every day, looking to understand novel human genetically derived drug targets and prioritize them and look for sources of validation for them and be studying kind of careful subphenotypes within, populations where that drug target may be playing a role and so on. And that could be a really differentiated data approach on the discovery side of the house. It could be a company that's building an extraordinary experimental data set to nominate novel drug targets. One of the companies in our portfolio, called Insitro, is building a very unique platform oriented around using induced pluripotent stem cells, iPSC cells, to create controlled environments where the genetic perturbation can really be studied, to say, well, if we make this genetic perturbation, what happens to the phenotype of the cell, and how does that subsequently interact with a potential drug that could ameliorate the disease state? So there's lots of really exciting approaches happening on the discovery side. Maybe even in between kind of target discovery and development, there's a lot of data sets that are now being brought to bear on drug candidate discovery and optimization. So you could imagine having a data set that describes high throughput small molecule screens, or from, let's say, a DNA encoded library platform, and really building a data set that allows you to learn which features of a small molecule correlate with which functional perturbations of those molecules, or binding properties of those molecules. And you could have a really kind of unique data set that guides how you discover molecules. And then on the development side, lots of ways too, right? So, going back to the polygenic risk score discussion, lots of different data sets could provide really interesting ways of stratifying the population that you're hoping to treat. And in so doing, could enhance the effect size that you get to observe clinically of a candidate drug. So, I hope we'll see more clinical trials that are leveraging things like polygenic risk scores to determine who might be the right candidate, let's say, for a preventive therapy or even for a disease-modifying therapy.
Absolutely. Those are all great examples. Maybe this is a good opportunity to transition into the next 10 years. What are some of the areas that you're most excited about now and going forward? And in particular, any that are maybe off the beaten path that others aren't thinking about as much as you think they should be?
I'm particularly interested in exactly those. That the framework that I use is sort of what I just outlined on companies that can step change how they do discovery as well as development. I think we've had a little bit of a period where we've sort of been skeptical of target discovery efforts. And some people have argued that, well, we're in a target-rich environment in some fields, maybe in oncology, where we know enough targets and it's been a problem of drugging them and finding the right modalities to drug them. And I think we believe in that. And a large number of the bets in our portfolio are focused on novel modalities to drug maybe biologically de-risked targets. I do think, kind of a set of, maybe dark horse companies that are going to come up and surprise folks are going to have built really key capabilities around target discovery and are going to, sort of go back to the biology drawing board and say, what are sets of targets that we just haven't done enough work to characterize and prioritize and nominate as novel, truly novel biology that we could intervene on. And so I'm a little, maybe a little bit of a controversial position vis-a-vis folks, honestly, who've looked at the trail of sort of target finding companies having really struggled to produce really novel targets. My view is some of those are actually going to succeed and that we need to support their growth over the next decade.
Sometimes it just needs time, right? It's not that it's a bad idea. It's just a little early.
Yeah, exactly. That could be, companies that are really investing in single cell sequencing approaches for target discovery. That could be companies that are investing in, studying the function of previously unstudied cell types. That could be companies looking at classes of targets related to RNA function or epigenetic function and, classes of proteins that we just haven't fully understood biologically.
Okay, so I think we're going to see more and more of this, and it may cause us to think about breaking the paradigm between rare disease and common disease even more than we are today. If common diseases can be broken down into uncommon combinations based around polygenic risk scores, proteomics, or other scores that allow us to identify a group of patients that's similar and could be treated with a targeted therapy, I think we'll be looking at a treatment landscape and a biotech landscape that's really different in the next couple of years. I hope you enjoyed that trip down memory lane. As I mentioned at the top of the episode, these were all from the first 100 episodes. There's some real gems in there. So if you have some spare time and are up to date on all the recent ones, then I really encourage you to go back there. Like I said at the beginning, if you enjoyed this style of episode and you want to hear more supercuts of themes, we're thinking about maybe we do it for newborn screening, maybe we do it for all the biotechs that we've spoken to. Where are they 5 years later in some cases after we've spoken to them? What did they learn from that process? I would love to hear your feedback in any kind of style of episodes you'd like. So happy 200th episode. Thank you so much for taking the time to listen to this. And as always, if you have feedback, guests you'd like to hear from, or anything else you'd like to hear, you can reach us anytime at podcast@sonogenetics.com. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sonogenetics on Twitter, LinkedIn, or visit sonogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.